Hello everyone. Maharana Pratap Singh Shikshan Sanstha, Mumbai's Anandibai Raurani Arts, Commerce and Science College, Vaibhavadi organizes the guest lecture series on the topic of and inventions in the life sciences and the inventions in the mathematical sciences on the occasion of National Science Day on the 28th February 2021. The basically National Science Day is celebrated every year since 1987 in the memory of the Raman Effect, which was discovered by the one of the most important physicians, that is the Sir C.V. Raman. Okay, for this effect, the Sir C.V. Raman is awarded Nobel Prize in 1930. So, the Sir C.V. Raman is the first Asian persons to honor the Nobel Prize in the entire Asian countries. So, in the memory of that, the National Council of Science, Technology and Communication is asked to the Government of India to celebrate this day as a National Science Day. So every year the 28th February is celebrated as the National Science Day in the India. So with concert with that, in this year every year the DST, that is the Department of Science and Technology, decided the theme of the National Science Day, and in the year the 2021 the DST as the decided the theme of this National Science Day is the future of STI, that is the scope and impact of STI on the education, skill and work. So basically STI is nothing but the science, technology and innovations. So whatever the innovations invented by the science up to now, that what is the scope and what is the future of these inventions is to be studied in the year, and we know all about to that concerns. So for this, our college and the Science Association is organizes the guest lecture series on the topic of inventions in the life sciences by Mr. Rahul Patil, that is the head and assistant professor from the Department of Zoology, Balwant College, Vita, and the science and inventions in the mathematical sciences by Mr. Vijay Shinde, that is the assistant professor from the Department of Mathematics, Anandibai Raurani Arts, Commerce, Science College, Vita. So we are firstly thanks to these speakers to accepted our invitations and they speak on their expert topics. So we are firstly thank to that two guests who are share their thoughts about their topics, and we are also thanks to our principal sir, Dr. C.S. Kakade sir, and the team of Science Association. So in this guest series, we are started with the first lecture of Mr. Rahul Patil sir, and then we are going to be the Mr.
Good afternoon to all dear participants. I am Mr. Rahul Patil, presently working as Assistant Professor and the Head Department of Zoology at Rare Shikshan Sanstha's Balwant College, Vita. At the very outset, I would like to congratulate the Honorable Principal of Anandibai Raurani Arts, Commerce and Science College, Vaibhavadi for celebrating this National Science Day in this COVID-19 pandemic. Also, I would like to thank the coordinator of this National Science Day, my friend Mr. Sachin Patil sir, for inviting me as a resource person in this National Science Day. So dear friends, as we are aware that we are facing this COVID-19 pandemic, and as we know that the celebration of this National Science Day, it is not possible in the physical way. So dear friends, today we are gathered virtually to celebrate the discovery of great Indian physicist Dr. C.V. Raman sir. So on this day in 1928, he invented the Raman Effect. So by celebrating this day as a National Science Day, we show our dignity and respect to the famous Indian physicist for his contribution to the field of sciences. Raman effect explains the effect on the scattering of light when passing through the different materials. So dear friends, the main objectives of celebration of this National Science Day is to bring awareness to people about the importance of the science in daily life and to encourage them by popularizing science and technology. So every year, the day is celebrated with a particular theme. So dear friends, this year, the theme of National Science Day 2021 is Future of STI, Impact on Education, Skill and Work. So, as a student, as a teacher, it is our responsibility to honor a great man and his invention by contributing greatly to the field of science through the innovation. So, dear friends, regarding this, the topic is allotted for me is the invention in life sciences. So dear friends, as we are aware that there are the n number of list of the invention that has been takes place in the life sciences, and that has been changed the face of entire world. So dear friends, today we are focusing about some important invention in the life sciences, and mostly we are focusing about the genetic engineering, or we can say the invention that has been takes place in the DNA molecules, or we can say in the field of molecular biology. So without wasting time, so let us start with our today's topic. So this is the timeline of revolutionary biological discoveries, or we can say the invention in the biological sciences. So dear friends, the journey of invention was initiated in early 18th century. So in this century, we are aware about the scientists like Darwin's and the Lamarck, and they have stated some hypotheses, the theories regarding the inheritance and the evolution. And we are also aware about the Mendel's, or we can say the theories of the Mendel's in the inheritance, or we can say in the field of genetics. So dear friends, as day is going on, there are the many invention has been takes place in the biological sciences, like the invention of the antibiotics in 1928, the invention of the gel electrophoresis in 1931, the discovery of the HeLa cell line in 1951, so on. When we talk about the recent invention in the biological science, and that is the CRISPR technology, and we are aware that in chemistry near in 2020, there is a Nobel Prize awarded for the discovery of this CRISPR technology. So dear friends, by using this CRISPR technology, we can hack our DNA. We can do anything with our DNA. We edit our DNA. We can insert the gene of another species into the human DNA. We can produce, or we can treat the different diseases. We can alter the ecology of some species. We can say that we can transform the food technology, or we also said that we are able to edit our human genome. So this is the power of science, and we can say in the technology. But dear friends, as far as my knowledge is concerned, the first invention in the biological science that reveals the mystery behind the life of the organism, and that is the microscope. So dear friends, a tiny molecule, and the function of the tiny molecules, and we can say the structure of the tiny molecules is not possible to see in our naked eyes, but that work is done by the microscope. And dear friends, due to the invention of the microscope, we can find out the exact structure of these macromolecules and the micromolecules, as well as we are trying to find out their functions. So dear friends, as the discovery of, we can say that the history of discovery of the microscope that was initiated in the 1590, the two Dutch spectacle markers, that is the Gicharius and Janssen and his father, discovered the first compound microscope in 1590s, and the journey is going on. And recently we have invented a number of microscope that has been used in the purpose of the different researches. So dear friends, this is the a picture of the electron microscope, which gives the structure of the macromolecules. So dear friends, the another invention of the biological science is the production of the vaccines. So dear friends, as we are aware that presently we are fighting against this COVID-19 pandemic, and everyone everyone in this world know about the importance of the vaccines. So dear friends, the journey, or what is meant by the vaccines. So dear friends, a vaccine tricks the body immune system into producing the antibodies to fight a form of the virus that is harmful to our bodies. Then, if the person ever encounter the real and indigenous virus in the body, at that time the body is ready to prevent it from the harming to the any different types of the cells. So this is the basic concept of the production of the vaccines. So dear friends, the journey of the production of the vaccine was initiated by the Edward Jenner. They have inoculated a young boy in England and successfully prevented him from the getting a smallpox. So dear friends, the Jenner used a lancet. You can see here the procedure that used by the Edward Jenner. So dear friends, the Jenner used a lancet to scratch some infected materials from a woman with a cowpox. So structure of the cowpox is similar to the smallpox. So dear friends, this is a journey, or we can say this is the beginning towards the production of the different vaccines, and we have the n number of vaccines today. So this is the chart where the vaccines has been done in the different particular months or years in the humans. Then we'll talk about how that vaccines eradicated the common diseases. So this is the flow chart, or we can say the diagram shows that the vaccine plays important role in the eradication of different types of the common infected diseases in USS. So dear friends, the another important invention in the biological science or the life science is the production of the antibiotics. So dear friends, as we are aware that day by day the number of infectious disease has been decreased, or we can say decrease due to the invention of this type of the antibiotics. So dear friends, as we are aware that in early 1981 to the 1995, the death from the infectious disease is increased by 4.8% annually. So after the discovery of these particular antibiotics, what happened that the death rate caused by these infectious disease or infectious pathogens has decreased. So you can see here the diagrammatical representation of how the infectious disease has been aimed by the discoveries of different antibiotics. So the first antibiotic was discovered, and that is the penicillin. So dear friends, this is the number of we can say this is the list of antibiotics which is used to eradicate the different types of the diseases. Then another important discovery in the biological science is the we can say invention is the production of the CT scan or the we can say the computerized tomography. So I'm not going to discuss about the detailed history about the CT scan. So these are the father of the CT scan. They have got the Nobel Prize in 1979 for the contribution for the production of the CT scan. So dear friends, the common use of the CT scan is used for the examination of the chest, abdomen, pelvis, or we can also say that it also used for the severe injuries from the incidents such as a motor vehicle accident. It also used for the acute symptoms such as abdominal pain or difficulties in breathing. So these are the some uses of the CT scan. And then we'll talk about the another invention, that is the modern form of the CT scan. It is the MRI, that is the magnetic resonance imaging. So dear friends, this is the figure, or we can say this is the machine that is used in the MRI scanning. So this is about the history of the discovery of the or invention of the MRI machine. So he is the father of the MRI, to magnetic resonance imaging inventor. Then these are the some Nobel laureates who got the Nobel prizes for the discoveries or the contribution in the production of the MRI machines. So, dear friends, what can be diagnosed by an MRI scan? So, we can see here by using this MRI, we can scan the different types of the abnormalities in the humans and the different types of the biological things. So, dear friends, today we are focusing about some important invention rather than the previously we see here. So, my topic is allotted of is the invention in the life sciences, and today I'm just focusing about the one term, or we can say the one side of the molecular biology, and that is the genetic engineering. So, dear friends, the genetic engineering is the process by which the piece of DNA are transferred from one organism to the another organism. So, dear friends, we have invented many techniques like the transformation by the somatic cell fusion, or we can say the transformation by the microinjection method, or the we can say the transformation by the viral method. So by using these methods, we can able to transfer the one piece of DNA into the another organism. So we can able to transfer the human gene into the bacteria, from the bacterial gene into the plant cell, or human gene into the different mammals, or fish into the different plant cells. So this is nothing but the genetic engineering. And dear friends, I'm very happy to say that this is the future of the life sciences. We can do anything, we can produce anything in a very cheap way, or we can say we are producing a n number of products by using these methods that is called as the genetic engineering. So dear friends, when we talk about the genetic engineering, we must know about the some important invention in that particular genetic engineering. Is that the first key term used in the genetic engineering is the recombinant DNA technology. So dear friends, as we know that this recombinant DNA technology, or the RDT, is a DNA made from two different organisms formed by a laboratory method. For example, the human gene and the bacterial plasmid. So we recombinant the two different types of the genes from the two different resources, and we do different things, or we can see that that type of the recombinant is used for the production of the different types of the hormones, chemicals. Okay, so then we'll talk about the gene therapy. So dear friends, the insertion of a functional gene or a gene into the cell, tissue, organ to correct the genetic abnormalities. As we are aware that the many peoples from the world are facing, or we can say fighting against these different types of the genetic diseases, and these type of the therapies, we can say that gives a relaxation to that of that particular peoples. Then we'll talk about the gene cloning. It is another important innovation in the genetic engineering. The gene cloning is nothing but the production of the exact copies of a particular DNA sequence in order to develop a line of genetically identical organisms which contain the identical copies of the same DNA. Suppose we are interested in the particular gene, so we can make that multiple copies of that particular interested gene by using a method that called as the gene cloning. And for that, we are using different cloning vectors like lambda phage, like cosmid, like plasmid, like phagamid, like yak, yeah, like bat. So these are the some cloning vectors we are using in the procedure of the gene cloning. So dear friends, by using these all techniques, we can form a different transgenic animal. So what is meant by transgenic animal? If genetic material from the another species is added to the host, the resulting the organism is nothing but the transgenic animal. You can see that you can watch the Hollywood movies like the Spider-Man, like the Batman. So that is in future it is possible to produce the Batman, to produce a Spider-Man by using this technology, and that's why we can say here the importance of genetic engineering, and I can say that genetic engineering is the future of the world. So dear friends, this is very important as far as the invention is concerned. So as we are aware that the invention of the PCR is also important when we talk about the genetic engineering, because when we talk about the genetic engineering, we require the multiple copies of DNA, and that is possible due to the discovery of this PCR machine that is called as the polymerase chain reaction machine that was invented by Carey Mullis. So the PCR performs or we can say in the laboratory, these PCR techniques used to make a multiple copies of the segment. So by using this type of the different methods, we can engineer our gene, we can alter our gene, and that is called as the genetic engineering. So dear friends, the group of technique and the science of making changes to the genes of the plant, animals, we can say by removing or by inserting or by change in the gene in order to change one or more of its characteristics, that is nothing but the genetic engineering. So dear friends, let's see one by one what are the different evolutionary discoveries has been takes place in the genetic engineering. So dear friends, when we talk about the history of genetic engineering, we must know about the term that was coined by the scientist, that is Jack Williamson, in his science fiction novel, that is the Dragon's Island. So earlier we saw the definition of the genetic engineering that was explained by this great scientist, that is the Jack Williamson, in his a novel, that is the Dragon's Island, in the 1951. So dear friends, when we talk about the genetic engineering, the history of genetic engineering, we can give a more importance to the importance to the discovery of the structure of DNA, or we can say the double helical structure of DNA that was proposed by the Watson and Crick in 1953. But here we can also remember the contribution of the Rosalind Franklin and his colleague in the discovery of the crystallographic structure of DNA. So dear friends, in 1962, the James Watson, Francis Crick awarded a Nobel Prize for the discovery of double helical structure of the DNA. So in this figure you can see that the structure of DNA is double helical, and we can see that or we can say that this is the magic molecule of the every living things. Okay, so then we'll talk about in 1972, the Paul Berg created the first recombinant DNA molecule by combining a DNA from the a monkey virus, that is a simian virus 40, with that of the lambda viruses that called as the bacteriophage. You can see here the a figure of that particular bacteriophage. So in 1972, the first recombinant DNA was produced by the Paul Berg with the fusion of the monkey virus or the simian virus 40 in the monkey with the lambda bacteriophage. In 1973, the Herbert Boyer and Stanley Cohen created the first transgenic animal by inserting the antibiotic resistant gene into the plasmid of E. coli bacterium. This is another remarkable invention towards the production of the antibiotics, or we can say the production of the different types of the antibiotics. So in we can say in early 1980s, there are different types of the invention has been takes place, and most remarkable invention in the 1980s is the invention of the PCR machine. So dear friends, you can imagine that in a minute, or we can say in a fraction of second, we can produce a million copies of our interested DNA, and just it is just possible because of the invention of the PCR that was carried out by the Carey Mullis, and he also awarded a Nobel Prize for the invention of the PCR. Then the another invention in early 1980s is the development of genetic fingerprinting. So dear friends, as we are aware that these techniques are used in the various purpose for the finding out the offsprings. It also used in the finding out the different criminals. So it is also used in the forensic sciences. So dear friends, this is another we can say remarkable invention in 1980s. Then the first transgenic mice is produced that carries the human gene. You can see here in this figure, these are the first transgenic mouse that carries the human gene that is called as the growth hormone gene. So you can see here the difference between these two animals, and that's why we can see that the genome is altered in that particular transgenic animal. So another invention in the early 1980s is the production of the transgenic plant by the we can say that resist against the different types of the herbicides. So this is the we can say this is the machine that is called as the PCR machine. Okay, then we'll talk about some important discoveries related to the genetic engineering, like the first GM or the genetically modified plants or organisms is used to make the chymosin, an enzyme used to making the hard cheese. So first gene therapy trials in human that is also carried out in early 1990s or the in 1990s. So dear friends, in 1996, the birth of first clone animal, that is the dolly, was takes place, or we can say announced, and that is the revolutionary, we can say remarkable innovation in the early 19th. So dear friends, when you talk about the history of genetic engineering, so when you talk about the era of 21st century, and the most remarkable discoveries or invention in the 21st century is nothing but the production of or the finding out the CRISPR technology, or we can say the CRISPR-Cas9 technology. So dear friends, with this technique we can do anything with our DNA. We can hack our DNA, we can edit our DNA, we can treat our DNA, or we can treat our different genetic diseases, we can alter the ecology of different species, we can transform the genes in the different types of the plants so as that particular plants produce more food, so we can edit our human genome. Okay, so this is the potential, or we can say this is the ability which is present in this CRISPR-Cas9 technology. And I'm very happy to announce that the Nobel Prize for the 2020 is awarded to the Emmeline and the Jennifer for the discovery of the method of the CRISPR and the Cas9 technology. So dear friends, a subsequent generation of the genetic engineering techniques that emerged in the early 20th century centered on gene editing. So the gene editing based on a technology known as a CRISPR-Cas9 technology that allows the researcher to customize a living organism's genetic sequence by making very specific changes to its DNA. So this is about the revolutionary the invention in the 21st century. So dear friends, the first we'll talk about the first invention, or we can say the first method is used in the genetic engineering, and that is called as the recombinant DNA technology. So when we talk about the recombinant DNA technology, we are using the two different sources. So we are fusing our interested DNA by the combining the two different types of the DNA. So dear friends, for that we require the two different types of the species, like the bacteria or the animal cells, where we are interested to make the recombinant piece of that particular DNA. So dear friends, by using different types of the cloning vectors, you can multiply it, or you can form some multiple copies of this recombinant DNA. So by using this method, we can uh, we can introduce the gene for the pest resistance, we can introduce a gene for a toxic cleanup bacteria, we can produce the different types of the transgenic animal, or we can produce the different types of the proteins for the stone washing of the genes, or also we can produce the different types of the proteins that dissolves the clot. So that's why, as I told you, that the genetic engineering is the life of or we can say the most important things for the as far as the human life is concerned. Then we'll talk about the some important tools required for the recombinant DNA technology. And here we can see that the enzyme, or we can say discovery of the restriction endonuclease, which helps for the production of the different types of the recombinant DNA technologies. So dear friends, as we are aware that the in prokaryotic nucleases that recognize the specific DNA sequence and cleaves the sugar phosphate backbone either within or close to the recognition site. So dear friends, the bacteria having a system, a particular immune system to fight against the virus infection, and that is done by the different types of the restriction endonuclease. This type of the restriction endonuclease cleaves the viral DNA and kills the virus by breaking the viral DNA at the specific site, and that's why we called as the restriction endonucleases. Okay, and that is called as the palindromic sequences. Okay, so a restriction enzyme is a protein that recognizes a specific short nucleotide sequence and cut the DNA only at the specific site, which is known as the restriction site. So this is called as the target sequence. So dear friends, till date more than 400 restriction enzymes have been isolated from these bacteria and that manufacture them for the use of the human purpose. So dear friends, after the invention of the restriction enzymes or the restriction endonuclease that are used in the we can say the different techniques of the molecular biology. So when we talk about the discovery of the restriction enzyme, we we can find the different types of the scientists that contribute their research in the production of or the in the synthesis, or we can say the finding to find out the restriction enzymes. And the first scientist is called as the Warner Arbor. He got the Nobel Prize in the physiology or the medicine in 1978 for the in vitro restriction of the lambda phage DNA by E. coli extraction. Then we'll see that the Hamilton Smith also got the Nobel Prize in medicine in 1970 physiology and medicine in 1978 that isolate and characterize the first restriction endonucleases, that is the endonuclease R, laterally we called as the HIND2 or the HIND2 molecule. So dear friends, this type of the different scientists contribute their research to find out the different types of the restriction enzymes. So the Daniel Nathas and the Dana contribute their research, or we can say they are the pioneer for the application of the restriction enzymes. So they have find that the exactly application of the different restriction endonucleases, and they have got the Nobel Prize in physiology and medicine in 1978. So dear friends, as we are aware that when we are initiate the recombinant DNA, for that we require to produce the more clone of that particular recombinant DNA, and for that we believing on the cloning vectors. So dear friends, the genetic vectors are the vehicles for delivering a foreign DNA into the recipient cells. So dear friends, these are some examples which act as the cloning vectors. So this is the PBR322. It is the cosmid, that is the super cos1. This is the phagamids, or we can this is the bacteriophage. There is another cloning vectors like yak, like bat, like sac. So these are the some cloning vectors as used in the transformation or we can say delivering the foreign DNA into the recipient cell. In molecular cloning, a vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into the another cells where it can be replicated and or we can say expressed. So when we talk about the some characteristics of the vectors should be small in size. They have a particular more than one restriction site. They have the some antibiotic resistance region. So these are the some characteristics of the good cloning vectors. So these are the some examples of the cloning vectors. Then we'll talk about the invention of the PCR machine that already told you that in a very short period of time we can able to produce a million copies of our interested DNA just because of discovery of a machine that is called as the polymerase chain reaction machine. So that was discovered by the Carey Mullis, and for that he got the Nobel Prize in 1993 in chemistry. So dear friends, I'm not going to talk about the detailed procedure for the amplification of the DNA. So let's switch on the next part. So suppose if we have all these type of the techniques, all these type of the machines or the technologies with us, then we are able to produce a genetically modified organisms, and that is called as the GMOs. So dear friends, as we know that the first genetic or the transgenic animal was produced, and that is called as the super mouse, that is created by the Ralph Brinster and Richard Palmiter in 1982. It was created by inserting the human growth hormone gene in that particular mouse genome. So the offspring was much larger than the parent. So you can see here the figure of the first transgenic uh, mouse. Okay, so that was formed by insertion of the human growth gene. Here you can see that this is the transgenic mouse, or we can see here the difference between the parent and this transgenic mice. Okay, so dear friends, as like the mouse, we have produced a n number of different transgenic animals like pig, goat, cow, sheep, fish for the yeah, we can say for the purpose of or the use of the different mankind's. Then we'll talk about the production of the transgenic mouse by the different techniques. So as we are aware that we are producing the different types of the transgenic animals by using some different types of the techniques like the somatic cell fusion, micro injection, or the we can say retroviral method. So these are the some methods used for the production of the different types of the transgenic animals. So in the past 20 years, the DNA microinjection has become the most widely applied method for the gene transfer in animal. So dear friends, introducing a transgene DNA directly into the zygote at an early stage of the development. Here there is no requirement of the vector has been takes place, and that's why we can say that the microinjection method was widely used for the production of the different types of the transgenic animal. So dear friends, when you talk about the procedure of this type of the production of the transgenic animal, so first we transfer of a desired gene construct from the another member of the same species from the different species into the pronucleus of a reproductive cells. Then we go for the in vitro culture of the manipulated cell to develop to a specific embryonic phase. Then lastly, we transfer the embryonic cell to the recipient female. So in the next figure, you can see the exact procedure for the production of the transgenic animal by the micro injection method. So suppose this is the interested mice from where we can generate our transgenic mice. So for that purpose, the first that mouse is injected with some sort of the gonadotropin that is called as the follicle stimulating hormone and the SCG hormone that create the super ovulation in that particular female mouse. Okay, and that is called as the that create the pseudo pregnancy. Okay, so dear friends, these type of the super ovulated mice are then mated with the different types of the males, and finally they sacrifice. Okay, so after 12 hours of the fertilization, we can collect the fertilized oocytes, or we can say fertilized oocytes flushed from the oviduct, and that are used for the production of the transgenic animals. You can you can easily differentiate the methods of the production of the transgenic animal by this figure. So these are the two different types of the mice that already we see that here. So when we made these two type of the mice, we are produce here the fertilized egg. Okay, and finally what happened that we just micro inject the foreign DNA into the pronucleus. For here, I just told you about this micro injection method, because we used some type of the micro needles here to directly insert the interested gene into the pronuclei of that particular the egg, fertilized egg of that particular mice. So we can see here the procedure used for the micro injection. So dear friend, this is the egg, and here we can use the micro needles. Here use the folding pipette so as to hold that particular egg. So by using this micro pipette, we can easily insert our interested DNA into the pronuclei of the fertilized egg. And finally, we can implant that fertilized egg into the pseudo-pregnant mouse to find out the different types of the transgenic animal, or we can say to produce the different types of uh, the transgenic animal. So dear friends, when we inject this particular fertilized egg, or we can say the modified egg into the pseudo-pregnant the male, after the completion of the pregnancy period, you can see here there are some transgenic animals or the transgenic mouse in the newly born mice or newly born young ones. Okay, so we can analyze from this the which mice are the transgenic. For that, we use the another method that called as the DNA analysis by using the PCR technology. So this is about the production of the transgenic mice by the micro injection method. Then we'll talk about the somatic cell nuclear transfection method is used for the production of the dolly. So dear friends, the dolly was the female fin dorset sheep and the first mammal born from the adult somatic cell nuclear transfection. So dear friends, the dolly was born in July 5th, 1996 at the Roslin Institute of Edinburgh, Scotland. The name Dolly comes from the suggestion by the stockman who helped her with her birth in honor of Dolly Parton because it was a memory cell that was cloned. So her birth was announced on February 22nd, 1997. The sheep was originally codenamed is 6LL3. So as I told you that the dolly is produced by the somatic nuclear transfection method. So for that we are using two different types of the sheep here, that is the adult fin dorset aves and adult Scottish blackface aves. Okay, so dear friends, these adult Scottish blackface aves is act as the egg cell donor. So by injecting some type of the hormones, we super ovulate that particular the female egg, and finally we collect the some healthy eggs. Okay, and finally by use of micropipette, we remove the nucleus of that particular egg. Okay, and that is called as the enucleated egg cell. Okay, so by using some sort of the micropipette, we will or we can say we remove the nucleus from this egg cell, and the cell that without nucleus is called as the enucleated egg cell. So meanwhile, from the adult fin dorset, we collect the some somatic cells that is used from that is collected from the adder cells, or we can say the mammary gland of that particular adult dorset ewes. Okay, so what happened that the that particular cells is cultured in the laboratory condition. They also shows different types of the abnormalities. So when that particular healthy cells are collected, and finally we are fusing that particular healthy cells or the healthy somatic cells with the enucleated egg cells by the process of electric shocks. So what happened that when that particular somatic cell is fused with that particular enucleated egg cells, for that we are using here the electroporation method. We use a small electric current so as the plasma membrane of the both cell is become fused, and the nucleus from the we can say fin dorset is transferred into the enucleated egg of the Scottish blackface apes. And finally, we also again give some electric impulses so as the fertilization has been takes place, or we can say the cleavage has been takes place, so as the cell division is initiated, and finally the embryo is formed, and finally we implant the embryo into the particular surrogate mothers. Okay, and finally, after the completion of the pregnancy period, that particular surrogate mother gives the birth, and that is nothing but our dolly. But dear friends, as we are aware that these type of the genetically modified organisms is not live a more years, because we have altered their genome. So we can see that in near about 5 to 6 years, that particular dolly was dead due to different types of the diseases and the abnormalities found in the that particular dolly. So dear friends, by using this transgenic production of the transgenic animal, we have produced the different types of the transgenic animals like the super pig. So we have know about the Superman. So as like the Superman, we have produced the super pig. As we are aware that in Marathi we can say that the गरज ही शोधाची जननी आहे. That means, as we are aware that day by day the population of world has increased, and to fulfill the demand of that particular population, we are using this transgenic animal. So dear friends, this type of the super pigs are the fast growing and lean animal that consume less food than the normal pigs, and they are fastly growing. Okay, so that means by microinjecting a gene that express the bovine growth hormone, so we can able to produce a fast growing pig. But as we are aware that, as I told you that this type of the genetic alteration is dangerous for that animals, and when we are going to produce the super pigs, that is suffered from the many health or the injurious health problems like the kidney and the liver problem, gastric ulcers, degenerative joint diseases, and the different types of the heart diseases. Okay, so dear friends, this is about the production of the another genetically modified organisms, that is the fish. So use of G to make a fish, cows for the to grow faster. Okay, so as I told you that to fulfill the demand of the or we can say the need of food, we can produce some sort of the different animals like fish, cows, uh, that will be a grow faster in the than the normal fish. Then we'll talk about the another example of genetically modified organisms, that is the silk milk goat. Okay, so this goat is formed by the injecting the silk production gene from the spider into that particular goat, and this silk milk could then be used to manufacture a wave-like material called as the biosteel. So as we are aware that the tensile strength of the we can say the silk of the spider, and recently we know that these type of the cells are used for the production of the surgical threads. They are used to production of the threads of the parachute. They are using the production of the bulletproof jacket. So dear friends, we can understand that this tiny spider is not it is not possible to provide that much type that much amount of silk to produce such type of the products. So reveal that we use this technique to produce the transgenic animal. So when we are injecting that particular silk gene into the other cells of that particular sheep, and that sheep is now is started to produce the silk in their milk. Okay, and that's why this is called as the silk milk goat. Okay, and that is used for the production of the silk. Then we'll talk about some another example is that these are the some transgenic fishes is used to study the development of the muscles, like the source of proteins. Proteins also use the some different types of the fishes like the pet fishes, or also called as the glowfishes. So we are producing these glowfishes by injecting the transluminant gene from the jellyfish into the this type of the fish, so as they are glowing into the different colors, and they're looking very we see here they are looking very good in the particular pot of the fish. Then we'll talk about the another important genetic engineering in the agricultures. So as we know that the using the genetic engineering techniques widely today to the produce genetically modified crops, or we can say the different plants. In most cases, the aim is to produce a new trait to the plant which does not occur naturally in the species. So dear friends, the main aims for the production of these genetically modified crops that are includes the resistance of the certain pests, different diseases or the environmental conditions, or reduction of the spoilage, resistance to chemical treatment, that is the resistance to the herbicides, or we can say the improving the nutrient profile of the crop. So dear friends, so this type of the we can say aim is behind the production of the different types of the genetically modified plants. So dear friends, these are some examples of the genetically modified organisms or the modified plants. That is the flavor saver tomato, which is formed in 1922, which has a long shelf life. Then in 1995, the BT potato was approved safe by the Environmental Protection Agency with BT protein, which is resistant to different types of the pest. Then these are the some list of genetically engineered plants, like the apples used for the non-brewing or we can say used for the food purpose. Then genetically modified potato reduce the bruising and the black spot. Non-brewing loss acrylamide used for the food. Then this is the field corn or genetically modified field corn used for the livestock and the poultry feed, used for the production of the fuel like ethanol, use the production of the high fructose corn syrup and the other sweeteners, or the corn oil, starch, industrial uses. Okay, so these are the n number of genetically modified organisms. They are used in the world, or we can say they are used in the US today now. Then these are the some examples of the another examples of the genetically modified organisms, and that is called as the strawberry or the blueberry. So dear friends, as we know that the strawberry is resist against the frost. So that idea is come from the flounder is a fish that lives in icy seas. It has a gene that prevent it from the freezing to death. Okay, so this is the fish which is live in the icy seas, and which has been which has a modified genome which protect from the frozen activity. So dear friends, we just isolated the gene from the fish and inserted into the strawberry, and that that strawberry is resist against that particular we can say to the freezing responsibility or the freezing activity. So strawberries are soft fruit that can easily be damaged by the frost. Okay, and that is done by these type of the methods, and that is called as the transgenic method. So but here what happened that when we introduce that particular gene into the strawberries, the color of that particular strawberry has been changed and it become blue in color. Then these are the some examples of the roses that are produced by the transgenic animal. This is the blue rose, or we can say this is the black rose. But in future, we can produce a multicolor rose also by using this transgenic technology. So dear friends, we can see here in early 90s, one plant is produced that is called as the tobacco. In that particular plant, what happened that the genes from the firefly, which is important or which is plays important role in the glowing purpose in the firefly, that is inserted into the tobacco, and now the tobacco at the night time that tobacco is become glowing. So these techniques, by using these techniques, we can do anything. So this is very important as far as the molecular biology is concerned. Then we'll talk about the another invention is that some scientists have produced the a cabbage plant with the scorpion venom. They have injected certain genes that produce the venom in the scorpion into the cabbage, so as that scorpion is a bug-proof veggies. So dear friends, as we know that the scientists added a gene from the producing scorpion venom to cabbage plant to kill the pesky caterpillars that it eats the crop. So we can do anything by using this type of the techniques. So dear friends, so main purpose behind the production of the we can say the genetically modified organisms in the medicine, in the agriculture, we have already seen that the use of genetically modified organisms in the different fields. So let's see that here we'll talk about the use of that particular animals in the medicine field. So dear friends, this type of the genetically modified organisms are used in the treatment of the different types of the diseases. First, we'll talk about the production of the insulin. As we know about that, the entire world is having with the diabetes or suffering from the diabetes, and as we know that the human insulin produced through the gene since 1982. So human insulin gene inserted into a bacterium, that is E. coli, to produce the synthetic human insulin for the treatment of insulin dependent diabetes. In past, what happened that the insulin was obtained from a cow or the pig pancreas that has a many problem. But it is possible that, but today it is possible that we can inject that particular human insulin gene into the bacteria, so as that bacteria is going to produce the insulin, and that insulin is used to treat the different types of the diabetes. Then we know that the producing the human growth hormone to treat the growth retardation, that is the dwarfism, the production of the polystim injection that contain the FSH hormone for treating the infertility, making human albumin, anti-hemophilic factors, and many other drugs, or we can say the other biopharmaceuticals under development through the genetic engineering, including the anti-cancer drug and a possible vaccines for the AIDS, malaria. So as we are aware that there is no vaccines on the AIDS, there is no vaccines on the malaria or many diseases, but these type of the techniques can help to produce the vaccines against the AIDS and the malaria. Then we'll talk about the applications of the G in the medicine for the production of the vaccination. So dear friends, as we are aware that today the microorganisms such as yeast is used to produce a virus antigen used as a vaccines that stimulate the human immune system against the different viruses. This procedure has been done successfully for development of vaccine against hepatitis B virus HBV that is now widely used. So dear friends, the genetically engineered vaccines hold a great promise for the future, like the G vaccines may be useful to prevent disease that have resist to traditional vaccination, including HIV, tuberculosis. More recently is the different types of the COVID vaccines or the corona vaccines like Covishield, and the different types of the COVID vaccine that are now inventing or presently inventing in the world. Then these genes are also used in the gene therapies also. So as we are aware that the most of the peoples from the every corner of world is suffering from the different genetic diseases, and as we are aware that there is no drugs or there is no medicine on the genetic diseases. So dear friends, these type of the technologies can give the relax to that particular genetically disorders. So by using this type of the gene therapies. So dear friends, the somatic gene therapy is designed to introduce a functional gene to body cells. So as we are aware that one genetical disease is formed due to the damaged function of that particular gene. So we can alter by adding a normal gene into the particular cell of that human. Okay, so this will cause a treating individual by targeting the therapy to body cell such as bone marrow or the different types of the blood cells. So gene therapy has been successfully used to treat a chronic lymphocytic leukemia, that is CLL, is another cancer, and also used for the treatment of the Parkinson disease. So dear friends, gene therapy is also being tested as a treatment for the cystic fibrosis, skin cancer, breast cancer, brain cancer, and even though in treatment of AIDS also. So however, most of these treatment are only partially successful. The major reason for this failure is insufficient vectors. Okay, so dear friends, recently the many scientists have discovered, or we can say that they are trying to produce some efficient vectors to transport such type of the genes into the patient or the cells of the different patients. So dear friends, in the future, as more efficient vector are engineered, the gene therapy is expected to be a common method for treating a large number of genetic disorders. So more that we can say that in upcoming future, uh, we have some sort of the chip in our bodies. So as we know that in our mobile there is a SIM card, as like a SIM card. In a future day, we have got a SIM card which provide proteins to our bodies, which are provide different antibiotics to our bodies, which will provide the we can say immune response to our bodies. So it is possible just because of these genetic engineering technologies. So dear friends, the next part of the gene therapy is that germline gene therapy. It is changing the gene of embryo used in the IVF before they are implanted. Suppose if you are found that the some genetical abnormalities in the embryos, so we can alter the gene of that particular embryos in the IVF technology before they are implanted into the surrogate mother. So these babies are designed to achieve a more desired look, skills, or talented such as a gender, hair, eye color, or intelligence, even though intelligence also. So that means we can produce a babies that we want. Suppose we want to produce a more intelligent baby. So you can produce the more intelligent baby by using this type of the gene therapies with the help of genetic engineering. So if the gene is inserted into the germline cell, such as a fertilized ova, it can be passed down to that person, that descendant. So application of the genetic engineering is made in the medicine is the n number of applications, and recently, as I told you, that the research has been going on to produce the different types of the invention in the genetic engineering. Then we'll talk about the use of genetically modified organisms as the xenotransplanters. So as we know that the transgenic animals that are genetically modified to have an organ that can be transplanted into the human, and that is called as the xenotransplanter. So how that are used to transplant the some organs in their bodies to the human, because they do not express a key foreign antigens, as we are aware that every cells of our bodies which has nucleus, and the nucleated cell having a certain sort of the MSC molecule. In human it is called as the HLA molecules, and due to the morphism in that, or we can say polymorphism in the HLA molecules, we cannot accept the organ from the different species. But such type of the organs has do not express any foreign antigens, so that's why there is a no immune response has been initiated, and finally that type of the organs is accepted by our cells, by our bodies. So this is the promising alternative to the human donors, as I told you that. So generally what happened that for the xenotransplantation, the pigs are used because when we talk about the physiology, it is most closely to the humans, and the we can say the pigs are less expensive than the other primates like the monkeys, or we can say the different other primates. So till date, what happened that the we are implanted certain types of these cells like the pancreatic cells, brain cells, red blood cells, eyes tissues into the human cell or into the human bodies from the pigs. But the trial of pig tissues transplanted into the human to treat diabetes, Parkinson disease, blindness are imminent, but the solid organ transplant like the heart, kidney, liver are still a several years away. So that means in future we can able to transplant the liver from pig to the human, a brain from pig to the human, or we can say the intestine from the liver, intestine from the pig to the human. So we can do anything by using this type of the genetically engineered technologies. So let's come towards the another point, and as I told you that this is the revolutionary discoveries or revolutionary invention in the 21st century, and that is called as the CRISPR technology. So as I told you that in 21st century, the two scientists has awarded as a Nobel Prize in chemistry for the invention in the CRISPR technology. So dear friends, as we are aware that in some bacteria have evolved a powerful system that called as the CRISPR to fight a viral infections. When a virus strikes a bacterium, that bacterium captures and stores a short identifying a sequence of that particular virus DNA, a sort of genetic memory card. Okay, so in our bodies we have a memory card. We store the our photos, we store the videos, as like that bacteria store some sort of the genes from that virus. Okay, and we can see that if the same virus attacks a future generation of the bacteria, they use the memory card to guide a killer enzyme to the identifier sequence in the new invader and cut it away. So scientists have cooperated this natural molecular machinery not only to turn off the action of the genes but also to insert new genetic code into the living organism. So dear friend, this is very interesting things now. So you can see here the exact method is used in the bacteria. So this is the virus and this is the bacteria. That bacteria, that virus is infected that particular bacteria by injecting their genetic material. So what happened that this bacteria is used, or we can say store some sort of the sequence in their memory, that is called as the genetic memory card. And if a new attack has been takes place, and that genetically memory guides that particular attack proteins or the killer proteins to kills that particular new, or we can say the same bacteria which infect after some days. So dear friends, as I told you that the scientists revealed that use of this technology for the different uses of the mankind, as like the treating diseases. So as we know that the genome editing technology is revealing which DNA sequence are evolved in diseases such as AIDS. We can use this technology by altering the ecology. So the spread of vector-borne illness like the malaria could be reduced by introducing a disease-resistant gene into the wild insect population. This technique is also used in the transforming the food. So CRISPR could be used to develop a drought resistance or otherwise the harder crops. The CRISPR mushrooms that don't brown have already been approved in the US. So the in US the we can say that the mushrooms has been produced by using this type of the CRISPR technology. So it is also used in the DNA editing of the human genome. Dear friends, the experiments with non-viable embryos shows that much work will be have to be done, and many questions answered before CRISPR can be used to edit the human genome. So in future we can use, or we can edit the human genome by using this type of the technology. So dear friends, as I told you that in future we can using this type of the technology, we can find different types of the remedies against the different types of the diseases or many problems. So this is about how the science and technology is useful for the mankind, or we can say useful for the different types of the living organisms present on the earth. So once again, I am thankful to the all dear participants for your patient listenings. So these are the some references that I used for the preparation of the today's presentation. So thank you. So once again, I'm thankful to the organizers of this National Science Day celebration, my friend Mr. Sachin Patil sir, and all the entire team of this Science Association for celebrating this National Science Day in this COVID-19 pandemic. So hope you have enjoyed this lecture. So all the best for your future. Thank you so much.
नमस्कार, मी प्राध्यापक विजय शिंदे. आज दिनांक 28 फेब्रुवारी 2021 हा राष्ट्रीय विज्ञान दिन म्हणून आपण सर्वत्र साजरा करत आहोत. दरवर्षाप्रमाणे याही वर्षी आपण राष्ट्रीय विज्ञान दिवस मोठ्या उत्साहात, मोठ्या जोशामध्ये साजरा करत आहोत. परंतु दरवर्षी जसं आपण विज्ञान दिवस साजरा करतो, वेगवेगळे उपक्रम राबवून आपण हा विज्ञान दिवस साजरा करत असतो. पोस्टर कॉम्पिटिशन असेल, मॉडेल प्रेझेंटेशन असेल किंवा व्याख्याने असतील, अशा वेगवेगळ्या उपक्रम राबवून आपण विज्ञान दिवस साजरा करत असतो. परंतु ह्या कोविडच्या महामारीमुळे आपण हा राष्ट्रीय विज्ञान दिवस हा ऑनलाईन पद्धतीने घेत आहोत. विद्यार्थी मित्र हो, ह्या राष्ट्रीय विज्ञान दिवस साजरा करण्यामागचा प्रामुख्याने हेतू काय आहे? तर ह्या विज्ञान दिन साजरा करण्यामागचा हेतू असा की विद्यार्थ्यामध्ये वैज्ञानिक दृष्टिकोन, त्यांना विज्ञानाची गोडी निर्माण व्हावी, अशा वेगवेगळा एक व्यापक दृष्टिकोन ठेवून आपण राष्ट्रीय विज्ञान दिवस साजरा करत असतो. तुम्हा सर्वांना माहीतच आहे की राष्ट्रीय विज्ञान दिवस हा थोर वैज्ञानिक C.V. रमन यांच्या रमन इफेक्ट ह्या संशोधनानिमित्त किंवा संशोधनामुळे त्यांना जागतिक दर्जाचा नोबेल पारितोषिक प्राप्त झाला आणि तेव्हापासून आपण हा राष्ट्रीय विज्ञान दिवस साजरा करत असतो. संयोजकांनी मला आजच्या ह्या विज्ञान दिनानिमित्त व्याख्यानासाठी जी थीम दिली आहे, ती थीम अशी सायन्स अँड इनोव्हेशन इन मॅथमॅटिक्स. मित्र हो, आजच्या ह्या स्पीचमध्ये, ह्या संभाषणामध्ये मी आपल्यासमोर मराठी आणि इंग्रजी ह्या दोन्ही भाषेमध्ये बोलण्याचा प्रयत्न करेल. जशी ही थीम दिली आहे, सायन्स आणि इनोव्हेशन इन मॅथमॅटिक्स. ॲज यू नो, देर आर सेव्हरल इनोव्हेशन्स हॅपन डे टू डे लाईफ इन एव्हरी डे, इन एव्हरी इयर. I'll definitely restrict myself when I am discussing the innovation of mathematics worldwide. I'll only discuss the innovations which is in India. As you know, there are several innovations which you can know. We have to notice all the innovations, but out of the innovations, I'll take a vital or more I feel personally these innovation makes differ from the others, or these innovations change the whole world in the mathematics or in science. मी असेच काही इनोव्हेशन्स आपल्यासमोर सांगेल मॅथमॅटिक्समधले की ज्या इनोव्हेशनमुळे गणितामध्ये एक खूप मोठा बदल किंवा खूप मोठा चेंज, किंबहुना सायन्स ह्या फॅकल्टीमध्येच ह्या चेंज आपल्याला पाहायला मिळेल. अशा इनोव्हेशन्सचीच मी आज तुमच्यासमोर थोडीशी माहिती सांगेन. फर्स्ट ऑफ ऑल, the first innovation in mathematics, that is zero. All of you know the zero is invented, the great mathematician astronomer Aryabhatta in the 5th century in India. And firstly, people don't know what exactly how to count or how to start the counting. After the invention of counting, they realize how to count and how to actual counting begin after the innovation of zero. Before, the mathematician struggles to perform the simplest calculations in arithmetic. Zero हा शोधण्यापूर्वी छोट्या छोट्या जे कॅल्क्युलेशन्स जी असायची ती कॅल्क्युलेशन सॉल्व करायला कॅल्क्युलेशन करायला तत्कालीन जे मॅथमॅटिशियन असायची सायंटिस्ट असायची ॲस्ट्रोनॉमर्स असायची दे स्ट्रगल फॉर द कॅल्क्युलेशन simplest calculations. But after the calculation, after the invention, after the innovation of zero, the calculation becomes more simpler. Nowadays, all of you knows what is the importance of zero, because without zero, right now we know that we cannot invent the computer, we cannot invent the further technology. So the invention of zero is quite important and milestone innovation in a mathematics in India. भारतामध्येच नव्हे, झिरोचा जर समजा शोध नसता लागला तर आज आपण जे वेगवेगळ्या टेक्नॉलॉजी वापरतोय, उदाहरणार्थ कम्प्युटर असेल, तुम्हा सर्वांना माहिती आहे कम्प्युटरमध्ये जी बायनरी लँग्वेज जेव्हा आपण वापरतो दॅट इज 0-1. So the system of computer goes in the 2 digit, that is 0 binary digit, that is 0 and 1. So the invention of zero is quite essential and important innovation in the world, in the mathematics world, in the science world. So one of the great scientists says the zero is widely seen one of the greatest innovation in human history, which is the cornerstone of modern mathematics and physics. Plus you can spin off the technologies. So I personally feel that zero is the most important innovation in mathematics. The second is algebra. As you know, very student feels the algebra is very complicated and they struggle to solve the algebraic problems or algebraic equations. Nowadays in India, in ancient India, the conventional mathematics termed as Ganitam. गणितम हा शब्द जो आहे तो गणितम हा एक संस्कृत शब्द आहे. Before the development of algebra, in the ancient mathematician or ancient mathematics in India, they use ganitam, the word ganitam, that is bij ganitam, which is known as bij ganitam means the other mathematics. Bij means another and ganita means mathematics. So this term is used in ancient mathematics before we know in the worldwide. So in India around 5th century, the system of mathematics that made astronomical calculation easy was developed. So particularly the whole algebra is used by the astronomers. So in those times the application was limited up to the only astronomy. जितके फक्त ॲस्ट्रॉनॉमी मध्ये जे काम करणारी जी वैज्ञानिक होती तितकीच मंडळी जी आहेत, तितकीच वैज्ञानिक जी आहेत ते ह्या अल्जेब्राचा यूज करायचे. And that is they are pioneers, means they are developed the whole algebra in ancient mathematics. The astronomical calculations are more complex. They involve many variables, they have derivations, they have much more calculations, but by using some algebraic methods, they are shortcut methods. By using the algebra becomes more simpler and easy. So I personally feel the second algebra is most important innovation in India. हा आता अल्जेब्रा म्हटलं की आज तागायत आपल्याला माहिती आहे अल्जेब्रा इज वर्ल्ड खूप मोठा असं व्यापक आणि खूप इट हॅज व्हेरी स्कोप ऑफ लर्निंग इट इज so wide subject nowadays. There are various methods developed, various calculation, various derivation are introduced in the algebra. So the algebra is one of the important innovation in mathematics. The second important innovation in India, that is trigonometry. That is quite important because this trigonometry word goes back to the Greek period. The character of the subject started resemble in modern form only after the time of Aryabhatta. This trigonometry basically started from the Aryabhatta. From here it went to Europe and Arabs, and then come to several modification and comes its present form. म्हणजे हा जो ट्रिग्नोमेट्री जो कॉन्सेप्ट आहे तो आर्यभट्ट ह्या काळापासूनच आपल्याला पाहायला मिळतो. त्यांच्या वेगवेगळ्या श्लोकांमधून, त्यांच्या वेगवेगळ्या त्यांच्या पुस्तकांमध्ये, त्यांच्या ग्रंथामध्ये आपल्याला पाहायला मिळतं. म्हणजे भास्कराचार्य असतील किंवा माधवा असतील, अशा ह्या वेगवेगळ्या मॅथमॅटिशियनच्या त्यांच्या ग्रंथांमध्ये, आर्यभट्टांच्या श्लोकांमध्ये आपल्याला ट्रिग्नोमेट्रीचा उल्लेख आणि वापर केल्याचा आपल्याला ऐकायला आणि वाचायला मिळतं. आणि मग तो ग्रीकपासून अरब कंट्रीजमध्ये जाऊन तो पुन्हा अशा वेगळ्या नवीन फॉर्ममध्ये जसं साईन, टॅन, कॉस ह्या फॉर्ममध्ये आपल्याला आज तागायत पाहायला मिळतो. सो दिस ट्रिग्नोमेट्री इज क्वाईट important innovation in mathematics, which I feel. In the ancient time, trigonometry was considered as a part of astronomy. Basically, the trigonometry right now also they are using in astronomy the several concepts of trigonometries. So at that time, trigonometry is only used in the astronomy, but nowadays it is used in various sections of the science. So that is important. In ancient time, there are three functions were introduced in trigonometry by our ancient mathematician in India. That is Ja, Koja, and Ukramaja. So these are the these three functions were used in trigonometry in that time. The first one is, that is Ja, means one of the first function that is we used in ancient time, that is Ja, that is R sin A. R is the radius of the circle. Sin A is the angle subtended at the center at the origin. And the second is Koja is the second function in a trigonometry which was developed in ancient mathematician, that is R cos A. Right now we are using. And the third one is Ukramaja, is nothing but R into 1 minus sin A. This is another function which is developed in or invented in ancient mathematics. So these are the functions which already used by our ancient mathematician in India, and we can understand this innovation makes so much differ for in the current era. वेगवेगळे असे इनोव्हेशन्स, या ट्रिग्नोमेट्रीच्या वेगवेगळे फंक्शन्स जी तत्कालीन मॅथमॅटिक्समध्ये काम करणारी जी आपली भारतातील काही तज्ज्ञ मंडळी होती त्यांनी ह्या फंक्शनचा वापर करून वेगवेगळ्या ॲस्ट्रॉनॉमीमध्ये असेल खूप मोठ्या प्रमाणात वापर केला, आणि तो आज वेगवेगळ्या फॉर्ममध्ये आपल्याला इथे पाहायला मिळतो. त्याचा खूप वेगवेगळ्या ठिकाणी त्याचा ॲप्लिकेशन आपल्याला पाहायला मिळतं. सो आय पर्सनली फील द ट्रिग्नोमेट्री इज वन ऑफ द इम्पॉर्टंट इनोव्हेशन इन अ मॅथमॅटिक्स विच ॲट दॅट टाईम. The next one is decimal system and quadratic formulas. This is one of the another important innovation in a mathematics in ancient times. In the classical period of Indian mathematics, the important contribution were made by the Aryabhatta, Brahmagupta, Mahavira, Bhaskara II, Madhava, and Nilkanta Somayaji. These are the mathematicians. They use the decimal system, the decimal number system for solving their daily calculations. They use some quadratic formulas for solving various complicated equations. So they already we have all the proofs in their shlokas they already use. So this is also one of the important innovation I personally feel in mathematics they invented, or innovation in ancient mathematician. In the 7th century, the Brahmagupta found the first general formula for solving quadratic equations. The Brahmagupta, this is one of the greatest Indian mathematician. He already developed some general formulas for finding the quadratic equation. So this decimal number system at that time they introduced, that is Hindu number system, which was our ancient mathematician developed. So decimal system and quadratic formulas, this one of the important innovation in ancient mathematics, I personally feel. So the next one is Fibonacci numbers. This I quite impressed about these Fibonacci numbers. This is also a great innovation of mathematics in ancient India. The Fibonacci number sequence we already knows. It started from 0, 1 and adding up to the last two numbers before it. That is our Fibonacci numbers which we knows. It was firstly described by Virahanka Gopala and Hemachandra, which is written in their writing, that is Pingala. He is a novel, a book, or its collection of shlokas. In that shlokas, Virahanka Gopala and Hemachandra, they are the great Indian mathematician. They already introduced about the Fibonacci numbers. The next one is the measuring length. This is also a great invention of Indian mathematics. In Indus Valley civilization, they are using in the 15th century, made by Ivory, the rural found during the excavation reveals the amazing accuracy of the decimal subdivision on it. So in the Indus civilization achieved the great accuracy in measuring the length, mass and time. म्हणजे हा लेंथ, मास आणि टाइम मोजण्याचं सगळ्यात मोठं जे इनोव्हेशन जे आहे तर जो इंडस व्हॅली सिव्हिलायझेशन त्या तत्कालीन भारतातल्या ज्या काय म्हणतो आपण त्यांना जे सिव्हिलायझेशन होतं त्यांनी हा खूप मोठा तिथे त्याचं इनोव्हेशन आपल्याला त्यांच्या वेगवेगळ्या साहित्यामधून आपल्याला पाहायला मिळतं. So this is one of the great innovation in Indian mathematics. The next is the finding weight of mass of the object. That is also a great innovation in mathematics. Again, finding the weight of the object. This is also invented or make the innovation in the Indus Valley civilization. So these are the some important innovation in mathematics I personally feel. But ह्या व्यतिरिक्त सुद्धा असे कित्येक तरी असे इनोव्हेशन्स आहेत की ते इनोव्हेशन झाल्यामुळे आपल्याला जगातल्या वेगवेगळ्या गोष्टी नव्याने अजून पाहायला वेगवेगळ्या संधी किंवा वेगवेगळ्या वाटा मिळालेल्या आहेत. तर ह्या गणितातल्या मला वैयक्तिकरित्या वाटणाऱ्या की हे इनोव्हेशन झाल्यामुळे ह्या इनोव्हेशन वरती आपल्याला कित्येक असे नव्याने अजून चांगले इनोव्हेशन्स आपल्याला ह्याच्या आधारे आपल्याला सध्या पाहायला मिळतात. तर अशा वेगवेगळ्या इनोव्हेशन्स आपल्याला भविष्यात ही सुद्धा वेगवेगळ्या पाहायला मिळतील. आणि ह्या विज्ञान दिनानिमित्त आपण सर्वांनी वैज्ञानिक दृष्टिकोन, विज्ञानाविषयी असणारी गोडी आपल्या मनामध्ये जपावी आणि विज्ञानाचा एक विद्यार्थी म्हणून कायम विज्ञानवादी विचार आपल्या मनामध्ये बाळगूयात. पुन्हा एकदा तुम्हाला सर्वांना विज्ञान दिनाच्या खूप साऱ्या शुभेच्छा. धन्यवाद.